So we speak of oxidative phosphorylation. That's how we make lots of ATP. This you should remember. We can have another view, answering the question, why do we breathe? Why do we breathe? This could be the next hand question. Why do we breathe? We breathe because we want energy. We're doing work, even if we're sitting here, not doing that much work, we're still doing work, cellular work. You're breaking down ATP to ADP. And you have to recycle that ADP to ATP. That's what you need to do. That takes energy, and that energy is gained from the breakdown of organic molecules in stages, and ultimately in the mitochondrion. You have the mechanism of ATP synthesis, which involves oxidative phosphorylation. So you breathe in order to get the oxygen into your body and to get the CO2 that is produced out. That's why we breathe. So we can live on fats, carbohydrates, CHO, or proteins. They're broken down to their subunits. They can be further broken down for fatty acids. This is called beta oxidation, where units of acetate are taken off the fatty acids and it's gradually broken down. And the fatty acid is short, say 16 carbon atoms in length. This can happen completely in the mitochondrion. And the fatty acids are longer. You need a special system, and that's where the peroxisome is used. So the peroxisome is important for the breakdown of fatty acids that are longer than 16 units of carbon. So those acetates are all coupled to coenzyme A, and that is used in the mitochondrion to feed a process called the Krebs cycle. Carbohydrates are also broken down. Typically we form glucose, because our important carbohydrate that we have as a storage is glycogen, polymers of glucose. Glucose is released in the cytoplasm, and this is broken down in 10 reactions that we call glycolysis. We form pyruvate that enters the mitochondrion and is converted to acetate coupled to coenzyme A. So acetyl-CoA. Amino acids are broken down in a variety of pathways, ultimately resulting in the formation of acetate. That enters a set of reactions that we call the Krebs cycle. That happens in the mitochondrion, where CO2 is produced. In the meantime, NADH is charged. It captures the hydrogen from the acetate, and that is then fed into the electron transport chain. And everything has to happen at the same time, so everything is coupled, and this is how ATP is formed. An important thing to remember, when I say ATP formation, what I really mean is the recycling of ATP from ADP and PI. So that coenzyme is broken down by work and recycled by metabolism. The process by which this happens is called chemiosmosis. We'll see later how that works. And it uses a proton gradient, a PMF. Okay. So there's a summary in this class. We're now going to focus mainly on the breakdown of glucose. There's a summary of what happens. And I think all I'm going to say is you should know this. It's not fascinating to repeat this. I don't think it's hard to remember. Maybe one thing I do want to mention is at some point here in the Krebs cycle, there are two GTP formed, and from the cell's perspective, GTP and ATP is kind of the same. That terminal phosphate on GTP can be transferred onto ADP, so as soon as you have GTP, you can form ATP. So we might as well say here we form ATP. Okay. A couple of details about glycolysis and the Krebs cycle that I want to emphasize. So first of all, hey, Yippee, you don't have to learn the reactions for me. Why? I still cannot remember I could if I tried. But we don't have to. It's not important enough. What is important is that you know that every organism has glycolysis. Virtually every organism. It always happens in the cytosol, almost always. But for the organisms that we care about, it's true. And importantly, glycolysis is regulated by the need to make ATP. Really what it's regulated by is by the ratio of ATP over ADP. When that drops, then the first two enzymes of the first and the third enzyme of glycolysis get activated. We'll see that in a moment. So it's the ATP over ADP ratio that regulates how fast glycolysis runs. The glycolysis speeds up when that ratio goes down, when ATP demand is there. So in other words, you start to be more active, you do more work, the cells in your body have less ATP, glycolysis picks up. You need to know the summary here of all these reactions, definitely, as well as this. And you need to understand there's two phases. There's an energy investment phase and there is an energy yielding phase, which we see right here. So we have an energy investment phase. First, the glucose is activated by the addition of phosphates coming from ATP. So that's the first enzyme here, hexokinase. The kinase, remember, is an enzyme that adds phosphate from ATP. Hexokinase. Kinases the hexose, glucose, forming glucose phosphate. Then we have phosphofructokinase that adds another phosphate. And these are two uphill reactions. And they're heavily regulated allosterically in response to the demand for ATP. After the activation of the sugar, it is split here in what's going to be a big downhill reaction, I'm going to guess. And then we get into the energy yielding phase, where hydrogen is taken from our organic intermediates and put onto NAD coenzyme, forming NADH. So that's that reduction step. And then ultimately we form 2 ATP in 2 stages. So in total, 2 ATP are first consumed, one and then a second. Then we split from a 6 carbon. We split here into 2 3-carbon molecules in this area here. And then we have 2 times 2 ATP formed. So net we have the formation of 2 ATP. Now, this ATP, which is formed right here in glycolysis, is ATP which is formed by an enzyme that binds the substrate and it binds the ADP together and it transfers the phosphor. And that's called a substrate level phosphorylation. To distinguish that from the oxidative phosphorylation that happens later on in the mitochondrion. This reaction by itself is not dependent at all on oxygen. This is not oxidative phosphorylation, substrate level. You should know. Okay, and pyruvate has been formed as the end product of glycolysis. What is the fate of that? It enters the mitochondrion. So it goes through the porins in the outer membrane and then through a carrier protein in the inner membrane gets into the matrix. And in the matrix, pyruvate is first taken by the pyruvate dehydrogenase, which oxidizes it a little bit, removes the carbon, so you get the formation of CO2, and you get the removal of hydrogen, forming another NADH. So this time, since there are 2 pyruvates for each glucose, you get 2 NADH at this stage for each glucose. And we already had 2 in glycolysis. Okay, that leads to the formation of acetate bound to coenzyme A. And acetate is then entering a cyclic pathway where it reacts with oxaloacetate forming citrate. Therefore, we call this the citrate cycle, citric acid cycle. It's also known as the TCA, tricarboxyl. Can't say that. TCA cycle, or the Krebs cycle, named after Mr. Krebs, who was, I think, British. In the Krebs cycle, 3 more NADH are formed per cycle, and that's 2 per glucose, so 6 NADH per glucose. And we see somewhere there are going to be 2 GTP formed, which we can equate with 2 ATP. And there's another coenzyme reduced, an FAD coenzyme. And that's 1 for each cycle, that means 2 for each glucose. Okay, so this happens in the matrix, you have to remember. Okay. A little bit about the coenzymes, because we love them. So an intermezzo. There are coenzymes that are important in metabolism. And you remember coenzymes interact temporarily with enzymes. They are carriers of the group. ATP carries phosphate, we remember that. NAD, and it's really NAD+, carries hydrogen, 2 hydrogen to be precise, which we can write as 2 hydrogen plus and 2 electrons. 2 protons and 2 electrons is a complete hydrogen. Same for NADP+, and how these two do it, we'll see in a moment. Then we have FAD, that's a flavin group containing coenzyme. And here we see the business part of this molecule. You can see how this molecule here is reduced by the addition of 2 hydrogen. Here we have the hydrogens added. Oxidized form on the top, reduced form at the bottom. The third type of coenzyme that we care about is coenzyme A, and it carries fatty acids. In their structure, if we look at the structure of these coenzymes, they all look as if they're derived from ATP. They're all derived from ribonucleic acid supplements. Why is this significant? Remember, ribonucleic acid supplements have stereochemistry. They have a flat base that can be recognized. This is a very good system to serve as a key, a handle that can be grabbed by an enzyme, active site. And with the extra P in NADP+, you can distinguish as an enzyme whether you need to be involved in anabolic or catabolic reactions. Now, the fact that all of these guys are central in metabolism and they're derived from ribonucleic acid supplements suggests to biochemists like me that maybe very early on in the history of life on Earth that there weren't really enzymes that were protein-based, but that everything was RNA-based. There's evidence for that, even though it's limited, but this is one thing that is striking. So this is really the fact that the important coenzymes are essentially derived from RNA supplements definitely tells us that these RNA supplements were always important in metabolism. Okay. So here we have the reduction of NAD and how that works. So we see here we have, in reality we have here this ring structure on this group. This is a nicotinic acid derived group. And I can't say that word, so I'm not going. We have a positive charge here. So what is the reduction? The reduction involves the addition of 2 full hydrogen. That 2 hydrogen, we can think of 2 protons and 2 electrons. This ring here can accept an electron and then we can add a hydrogen there. That means we have a proton left. So for the full reduction, what we actually get, we convert NAD plus with a positive charge here to NADH with 1 hydrogen and then an extra proton. So I'm telling you the reduction of NAD really is NAD plus is converted to NADH and a proton. But the bottom line is it's 2 full hydrogen. So that's a reduction, and the oxidation is the reverse reaction. So where's the difference between NAD plus and NADP? That's the extra phosphate right here at this position, which is the 2' position. on my ribose sugar. Okay, now you should recognize ATP, I've already told you that. GTP is very similar, has a different base, you can recognize that. You should be able to recognize these guys. Easy points on the next exam, I can promise you. How are we doing? We're almost out of time. Okay, so NADH and FADH2 are formed in the Krebs cycle. What happens with that? These coenzymes donate their electrons to the electron transport chain. And there's a lot of energy in those electrons, remember, because it's almost like rocket fuel, remember? If we're going to allow those electrons to ultimately fall to oxygen, there's a lot of energy there. The electron transport chain sits in the mitochondrial inner membrane, and ultimately electrons go to oxygen, and in this process a proton gradient is formed. Later that proton gradient is used for the synthesis of ATP. So we see that energy is temporarily stored in an electrochemical gradient. And this is the majority of the energy that is released after glucose is broken down. Now, how much energy approximately an NADH H+ couple allows for approximately 3 ATP to be formed. It's a little bit less, we'll see later. FADH2 is approximately 2 ATP. The formation of that ATP in the mitochondrion, which depends on the presence of oxygen, we call that oxidative phosphorylation. All right. I think that's enough for you today.